With that, I'd like to welcome Bruce Beutler to the stage. Bruce is now at UT Southwestern after a brief sojourn in Stockholm last year, and we'll be talking on genetics and innate immunity.
Well, thank you very much. Very nice to be here. I want to tell you tonight just my view of the evolution and genetics of the immune system, kind of an overview. And then I'd like to tell you very specifically where we stand with using ENU mutagenesis as a tool to dissect immunity and to understand it mechanistically. And finally, I'll tell you the results of a couple of screens that illustrate just how we're able to understand some very complex processes using an unbiased genetic approach. I'd like to begin by considering just how well immunity protects us. And there are ways of looking at this question just by analyzing longevity and the causes of death among humans. And I borrowed this slide from Jean-Laurent Casanova, who published it a couple of years ago in the Journal of Experimental Medicine. If we look at longevity statistics in the United Kingdom in the year 2000, we see just about what you'd expect. The average person lives to be about 80 years old, and it's extremely rare that anyone dies before the age of 50. In fact, we consider that quite exceptional and usually a tragedy when it does occur. On the other hand, if we look at the situation in Mozambique, we see something a little bit shocking, that the average person dies in his or her late 30s, and it's extremely rare that anyone would live to be 80 years old. So obviously how long you live depends upon where you live in large measure. The next shock is that if we go to Liverpool, England, in the year 1860, we see that the situation was even much worse than in modern-day Mozambique. The average lifespan then was about 10 years of age. And we see again that there was an attrition extending across all of the lifespan so that virtually no one would live to 80. So we've had two surprises, we might say. The third surprise for me was that things didn't get worse if you went a bit earlier in European history, nor did they get worse if you went even to Neolithic or Paleolithic times, at least as the fossil record tells us. You might guess that this early attrition is due mostly to infection, and I'll show you that in fact it is. And that's the extent to which the immune system can protect us in the wild state. If we look today at the whole planet and we ask simply, what do people die of? Approximately 25% of all people who are born, 14.9 million people per year at present, die of an infectious disease. This is not the major cause of attrition in our species. You can see that cardiovascular disease slightly edges out infectious disease, but there is something special about infection, and that's the age at which it causes death. Here, in the developing world at least, you can divide up lifetime into 0 to 4 years, 5 to 14, 15 to 44, and so on. And you see that early on, by far, the major cause of death is infectious disease. If you sum the causes of death through the age of 44 years, infectious compared to non-infectious deaths, you find that infection is the most major cause of death through the reproductive lifespan of Homo sapiens. And on that basis, you can conclude that at present, this is the major selective pressure operating on our species. It occurs to us to ask how many genes offer us robust protection against infection. And the answer is that certainly there are a large number of targets. We know now of hundreds that we could simply name if we sat down to make a list. But we had an inkling that this was true even quite a long time ago from studies of heritability. It would be predicted that if the target size is large, if there are hundreds or thousands of genes with non-redundant function in immunity, then there would be a lot that could go wrong with immune function, and many germline mutations would cause some kind of vulnerability, particularly if there were semi-dominant alleles, as we know there are. This would result in a high heritability of immune failure, which we could define by premature death from infection. This was actually analyzed by T.I. Sorensen back in 1988, and he used a classical method to analyze just how heritable death from all different causes was. That was an adoption study methodology. In the year 1928, in the country of Denmark, 960 registered adoptions were performed, and Sorensen was able to follow both the adopted children and their biological parents across a period of 60 years and ask what each set died of. If he looked at all-cause mortality, as you can see, if we're separating the children into two groups here. If their biological parents were alive, then they had the mortality statistics shown by the gray curve. If one of their biological parents or more died of any cause prior to the age of 50, then the children themselves were more likely to die, and their relative risk of premature death was 1.98. The argument could be applied to all kinds of death, but I'm only going to show you the statistics for infectious disease death. If both biological parents survived and did not die of an infectious disease, then the children had mortality statistics as shown here. They themselves would very rarely die of infection. But if one parent had died of infection before the age of 50, then the curves were even much more separated than for all-cause mortality, and the relative risk of an infectious disease death was 5.81. This was the most strongly heritable form of premature death that Sorensen could identify, more so, for example, than cancer or cardiovascular disease, which we might have guessed would be less environmentally influenceable, but it seems not to be the case. We might also guess, because the children lived during the golden days of antimicrobial therapy, that if antibiotics had not been available to them, as they were not available to the parents, then perhaps their mortality would be even much greater, but we don't actually have those data. All we can conclude is that, in fact, premature death from infection is more heritable than premature death of any other kind that was analyzed. All this would say that, in fact, there are a great many genes that are necessary for the integrity of immune function. And that brings us to the question of just how so many genes were marshaled together to make our immune systems. We know something about that, of course. We know that some elements of the immune system are shared by animals and plants, and so they date back probably 800 to 1 billion years or so. We can guess that in the beginning, the first things that conferred resistance might have been antimicrobial peptides or even antibiotics, but it would be expensive to maintain a constitutive system for protection against microbes, and therefore sensing and signaling components of an immune system, a prototypic immune system, must have arisen quite early on to regulate immune responses. Some of those components clearly predated the split between plants and animals, among them the leucine-rich repeat domains of receptors, the tear domain, the nod-like receptor domains, and these are observed in virtually all multicellular life forms. I've tried to color code them to some extent here, so you can see the leucine-rich repeats of toll-like receptors in animals have their counterparts in plants. We know of kinases involved in immune signaling like IRAK4 that are similar to non-RD kinases of plants. We have the tear domain in both animals and plants represented in toll-like receptors and their adapter proteins. And we have proteins like the NOD-NALP family of receptors that are quite similar to nucleotide binding and tear domain containing receptors of the cytosol in plants. And all this can be taken as evidence that innate immunity has very, very deep roots in the phylogenetic tree. Something very remarkable happened about 550 million years ago with the Cambrian explosion. And I think all of you know about this. There came a time in evolution when the oxygen environment of the Earth was very rich, and there were warm, shallow seas, and fantastic creatures began to evolve, most of which don't exist in this sort of form anymore, but it was at this time that structures represented in most animals today first began to evolve, such as eyes and teeth and limbs and digestive tract and skeleton and so forth, and these were transmitted forward into later life forms. But of great interest in the late Cambrian period, there evolved a new kind of cell that was passed down at least to a lot of the vertebrates that live today, and this cell is the lymphocyte. The lymphocyte took origin in different structures. In jawless fish, we see that lymphocytes originate in the typhlosole and in gills. We see in mammals that the thymus and bone marrow are the source of these cells. Also, they arise in the fetal liver and to some extent elsewhere. And these cells, of course, mediate adaptive immunity. It's known that proto-B cells and proto-T cells antedated immune function in the sense that they're present both in jawless and in jawed vertebrates. And the key thing that made these cells useful in immunity was that RAG genes somehow were transmitted, probably horizontally from another source, and began to mediate recombinatorial events. The substrate on which they operated was different in both cases. So we know now that in lampreys and hagfish, we have leucine-rich repeat receptors that are recombined to give antibodies of a kind that have an enormous diversity. We know that in mammals we have immunoglobulins and T-cell receptors that also have enormous diversity, and this is the basis of adaptive immunity. The legacy of microbial predation then became the immune system, both the innate and the adaptive immune system, and the legacy of the immune system is autoimmunity. So wherever we see a disease, and these diseases are very common, that involves inflammation, anything that ends with the suffix itis, we may guess that there's some problem with limitation of immune function or with tolerance to self. And so it is that we have diseases like rheumatoid arthritis, gout, type 1 diabetes, systemic lupus erythematosus. All of these involve failure of tolerance or destruction of self tissues, in some cases more by the adaptive system, in some cases more by cells of the innate immune system. We could even look more broadly and we could say that a lot of diseases that we didn't used to consider inflammatory, now we do see as inflammatory, at least in a way. We could point to atherosclerotic plaques that are loaded with mononuclear cells, foam cells, and they definitely seem to have an inflammatory component. Furthermore, toll-like receptors seem somehow involved in the formation of atherogenic lesions. So the immune system certainly came to occupy a lot of the space of modern medicine. The inferences to draw then at the start are as follows. First of all, in recent millennia, infection has been the most powerful selective pressure operating on our species. That's absolutely clear. The selective pressure of infection, both recently and also in the very remote past, led to the allocation of a large fraction of our genome to defense against microbes. We can speak of hundreds to thousands of genes. It could very well be that several thousand genes at least have a non-redundant function in one form of defense or another. Although we don't think of ourselves as machines very often, we have in fact become machines dedicated to immune defense. But I'd argue that we're very imperfect machines in that regard. It becomes sort of a glass half full, glass half empty argument, but we can see that obviously in the wild state, immunity often fails us. Most humans live an average of about 10 to 15 years, and most mortality is infection-related. The interesting thing is that our brains, more than anything else, seem to have greatly augmented our immune defenses in recent years. And we can think of this as a kind of adaptation of evolution that occurred right before our eyes, within living memory, and it's dramatically changed our species. We haven't changed morphologically, but we've certainly changed in terms of survival. Our lifespan has increased about 8-fold on average since the last 150 years. And much of this has to do with the implementation of an evolved evolutionary prowess in thinking and reasoning and recording what we know, and all of this crystallized rather suddenly and unexpectedly. In fact, it's questionable just how much farther we can go, but certainly we can be optimistic that we can improve the human condition even more. Nonetheless, coming back to basics, we have to acknowledge that without immunity and in the wild state, we would never survive even to the age of 10 if we didn't have immunity. Even crippling a part of our immune system will prevent that. And we would never have developed the kind of intellect or culture that we need to extend our lives further. So the big question confronting us that we can answer and begin to approach genetically is, what are the genes that protect us from infection and just how do they work? I liken the problem to understanding the function of a watch, because I'm a mechanist and I believe that we really are machines, and our immune system is one large machine with disparate parts. I would ask you the question, if I showed you a wristwatch, could you really understand its workings, knowing nothing else but what you see from the outside? And the answer would be probably no. On the other hand, if you had a list of essential parts, could you understand the workings of the machine? And I would say yes, probably you could. You would have to do experiments. You would try to fit cogs and gears together, and you would try to understand how they work. And in that humans are watchmakers, you probably would be able to learn how the instrument operates eventually. And that would correspond to the kinds of experiments that we do all the time. But the list of parts is key, and that's what genetics gives us. It gives us the list of parts, and it's up to us to then decipher the mechanism of how each works. When I talk about genetics, I'm talking about classical genetics or forward genetics. This is a system by which one makes mutations at random to create phenovariants. Then one can go back and find the precise mutation that caused the phenotype that is seen. This approach has the advantage of being completely unbiased. It yields authentic discovery. Sometimes it can even uncover new phenomena that one didn't suspect were present in the beginning, all by creating exceptions to the norm. The main limitation of forward genetics is that there is redundancy. To some extent, our genes are covered one by another because they duplicated and differentiated over a period of time, and a gap can remain also between mutation and understanding mechanism. Practically speaking, what we've been doing for quite a long time has been to mutagenize mice with a chemical agent, an alkylating agent called ethyl nitrosourea. These mice are then inbred through a series of generations to produce G3 animals, which can be homozygous for the mutations produced in their great-grandparents. And in all, over a period of more than a decade, we've made 9,347 pedigrees of this type. We've produced 38,767 G1 males, and in all, well over 100,000 G3 mice for screening. And when you do this, you see a lot of phenovariants, just visible phenotype. You can imagine that you're also creating a lot of problems with the immune system, and if you screen intelligently, you can find a lot of exceptions where there's a failure of immunity in one aspect or another. How do you find the mutation once you've created a phenotype? This is done, of course, by positional cloning, and that used to be very difficult. It took several years to track down causative mutations. The first such mutation we cloned occupied the entire lab for a period of 5 years. Mapping was typically performed using thousands of meiosis to prove that the causative phenotype was between two closely spaced markers on a chromosome. Usually we would narrow the critical region to a size less than 1 megabase before going to search for genes. All of the chromosome was terra incognita, and so a contig had to be built across the region, and genes within that interval were detected by exon trapping, hybridization selection, computational methods like GRAIL, and eventually by random sequencing, shotgun sequencing with blast searching against EST databases. That's all something in the past now. Since the mouse genome was sequenced and released One never needed to make a contig again or search for genes again. Instead, once a critical region was defined, one would just go to a genome browser and see what the gene content was, and those were the candidates to be tested. It was still difficult to track down a point mutation in a critical region several megabases long, because typically there were 8 genes and perhaps as many as 60 or 70 exons per megabase, and a single operator might check only 1 or 2 genes per week. In my lab, U Shah wrote a program specifically designed to automate this process. What he did was to analyze the critical region with a computer. Once we got to a region that contained less than 1,000 coding exons, the region would be masked to hide common repeats. Primers able to amplify every coding exon would be constructed, and also a second list of primers for sequencing would be generated. And all of these would be ordered in 96-well format. A robot would perform the amplifications from wild type and mutant DNA. The products would be cleaned up, their optical densities measured, they'd be diluted, and the right primers would be added to the right amplification products, and you'd get another stack of plates that you'd put onto a sequencer. Then a computer would call the mutation, and either you'd find it or you wouldn't. If you didn't find it, it would tell you the degree of coverage, and a second iteration could be done to go back and try to find what you had missed. Even this got to be obsolete, although we did use it to find as many as 50 or so mutations in our best years. More recently, if one had a mutation and one was really sure that it was recessive, then you would simply take the whole genome of the mouse, whole genomic DNA, and sequence all of it, and then apply a series of filters to look only at coding region, because that's where phenotype emanates from with ENU. And eventually, by capillary sequencing, you'd wind up with a list of about 10 or so true mutations in a G3 mouse, and one of them would be the causative mutation. It would lie within the critical region. This too has become obsolete because whole exome sequencing in the mouse displaced it. Now it's possible to sequence about 20 exomes per machine per week, and one can do this in sufficient depth that, first of all, validation sequencing may be obviated soon at least. One can detect heterozygous mutations with high efficiency, and the new policy is to take all the G1 mice that are the grandfathers of the G3 mice, to bank their sperm and to sequence their genomic DNA. In this way, one can archive mutations, and also one knows up front what all the candidates are immediately if a G3 mutant is spotted. I've been doing this for some time now with Chris Goodnow and his group, and collectively our labs as of today have banked a total of 11,000 point mutations in 7,122 genes, and these are all archived and all searchable on our websites. Mapping also has gotten much faster. We used to map on huge numbers of meioses. We now map by a technique called bulk segregation analysis, using the black 10 strain of mouse, which is very closely related to black 6, so phenotypes don't disappear as the result of modifier loci effects. In the usual case, about 30 mice, half with the phenotype and the other half without, are used, and one pools the DNA from each and then looks for concordance between genotype and phenotype across all of the markers covering the genome, and this takes about $400 to map a mutation to a chromosome. And I'm afraid the computer won't advance anymore, so I hope somebody has a battery or another clicker. But advance it again. Okay, this is what bulk segregation looks like. You see here is a phenotype that we called AOBA. It's a mouse with chronic renal failure. You can find the mutation on chromosome 1, as you can see by this peak, and this turned out to be in an unconventional collagen locus. So, one question people ask me is how much has this massively parallel sequencing approach accelerated mutation finding? And the answer is, as of a year ago, before I moved to Dallas, we had a total of 59 phenotypic mutant strains that had been analyzed by sequencing with SOLID. Next. We found 29 causative mutations, and in 30 cases the mutations hadn't been found, but in only 6 of those had mapping been performed. And the interpretation is that whole genome sequencing often finds the obvious mutations immediately. Those that are non-obvious require mapping, which presently goes more slowly than sequencing. But still, newer methods are going to make mapping obsolete as well, and the expectation is that about 100 phenotypes will be solvable per year. I'm also often asked what kinds of mutations cause phenotype, and here too we have some statistics. I'll just point to the fact that of a list of 234 mapped phenotypes which have been solved in our lab, we can point to 211 mutations where there is clearly a mutation in coding or splicing sequence. In the 23 remaining cases, the situation remains an open question, but suffice it to say, we've never found a phenotype that wasn't caused by a coding or splicing error, and I believe that's the rule in all of the ENU literature. We can also say that about 68 frank null alleles were observed among these 211 mutations, and 134 either non-critical splicing errors or mere missense alleles were found. So for every frank null allele, there are about 2 phenotypes caused usually by a missense mutation. That's obviously null. That's where most of the phenovariance comes from. One might wonder also how many genes does ENU mutate to phenovariance in a given population of G3 mice, and what does it take to saturate? In other words, if we look at 1,000 G3 mice, is that enough, or 10,000? How much damage are we doing to the genome? It's possible to simulate mutagenesis now in silico and estimate how many genes have been destroyed in a given screening project. The assumptions that we make are, first of all, that there are about 60 coding and splicing changes per sperm that generates a G1 mouse, and this we know from having sequenced a lot of them. We also know the experimentally observed base pair substitutions that ENU causes, and that's factored in. We also know the ratio of nonsense and critical splicing mutations to missense mutations, and we know that about 21% of missense alleles cause detectable phenotypes. To give you one example of where such a simulation was performed, a postdoc had looked at 6,303 G3 mice, and we were able to estimate that this would test 42% of all genes for phenovariance. So it's not really a gargantuan project to look at a rather large fraction of the genome. Finally, many people wonder how often do we ever find anything new by this approach, and that's a valid question. Obviously, it varies depending upon what we're looking at. If we look at a very well understood phenomenon, we're likely to find less than if we imagine a screen for something that isn't understood at all. But over a period of time, at some point where we had accumulated 195 phenotypic mutations that had been solved, these fell into 138 genes, and 32 of them were of major novelty, as I would call it. 10 out of the 138 were somewhat novel in that they at least illustrated new things about the genes that weren't known, and the remainder were not novel. So about a third of the hits that we find are at least enlightening in one degree or another. I would like to tell you about 2 screens that ask 2 questions to illustrate how the ENU mutagenesis process works and how it can tell us about an immune process. These screens were very simple. The first was a life or death screen. We simply inquired what proteins does a mouse need to survive for 7 days following mouse cytomegalovirus infection. Wild-type mice all live when given the virus. We wanted to know what they need in order to live. The second screen asked simply what proteins are needed for a T-dependent or a T-independent antibody response, or for the formation of T cells or B cells, or both. The first screen was easy to perform because the dose-response curve of mice to mouse cytomegalovirus is so sharp. In other words, if mice are given 10 to the 5th PFU of the virus, they never succumb to the infection. And on the other hand, if they're given 10 to the 6th PFU of the virus, they almost all will die of infection over the course of 7 days following inoculation. So we're able to pick up subtle degrees of immune failure by giving them the low dose of virus and looking for animals that die. In all, we tested 22,600 G3 mice, challenging them with 10 to the 5th PFU of mouse cytomegalovirus. We found about one pedigree in 33 carried a recessive mutation that caused susceptibility, and in all we detected 48 mutations that were conditionally lethal during MCMV infection. Many of these were lost because we began when we didn't have the means to preserve germline material from the animals, but in all 23 of them were positionally cloned. And these are the 23 genes that were identified. You can see that they fall into categories of sensing problems, sensing combined with developmental problems, signaling problems, effector problems, pure developmental problems, or problems with cellular or cardiovascular homeostasis. And then it was incumbent upon us to try to put these together into a sort of a picture, a high-level view of what has to happen with the mouse innate immune system for it to survive infection with this virus. I stress that this is all a look at the innate immune system because a mouse with a RAG mutation will do perfectly fine through 7 days following infection. You don't need T or B cells to survive. When a cell is infected, it was already known at the time we started our screen that the NK cell is a critical responder for limiting the infection. And from the work of Sylvia Vidal and Wayne Yokoyama, it had been deduced that the virus produces a protein called M157 that interacts with an NK activating receptor called Li49H that drives proliferation of NK cells and also causes degranulation of the cell, leading to elimination of the infection. Our own screen soon showed that there were other requirements as well. Specifically, dendritic cells had to become involved to co-activate the NK cell, or the mouse would die. And double-stranded RNA or DNA of viral origin were important here because mutations affecting TLRs 3 or 9 showed up in the screen, and so too did a mutation in UNC93B, known as 3D. This protein is needed for these nucleic acid-sensing TLRs to reach the endosome where they function. Mutations called Pococuronte and Odiose and PenR2 showed up in TLR signaling proteins as well, those being MyD88, IRAC4, and IKB kinase gamma. And we also found a mutation called NDECA that affected IRF1. We found an allele of TRIF called LPS2 that was lethal. All of those mutations led to a dead mouse. We found, too, that in the NK compartment, interferon signaling was crucially important. Macro1 and Macro2 were tracked to both chains of the IFNAR receptor complex. And from the work of others, it had become apparent that the IL-12 and IL-15 receptors were crucially important. 2 separate alleles of STAT1 were picked up, and so we began to speak of sensing and signaling components of the response. We knew of effector mutations as well. Many of these caused hypopigmentation as they not only affected the apparatus for degranulation of the NK cell, but also prevented transfer of melanin granules from melanocytes to the hair shaft. Not all of the mutations were in that category. For example, JINX a mutation in UNC13D was strictly involved in hematopoietic cells and wasn't part of the other pigmentation apparatus. An unexpected mutation called Mayday and a later allele called SOS showed up in the screen. Both of these mutations were in KIR 6.1, one component of an ATP-sensitive potassium channel that was located in the vascular endothelial cells. And it appears that these mice, which die very suddenly and early on following infection, are dying principally of myocardial ischemia or arrhythmia. In other words, these proteins are not needed for the cytokine response per se, but they're needed to survive the cytokine response. And those mice are also killed very easily by tiny amounts of lipopolysaccharide. There were developmental proteins too. For example, Sphinx was a membrane-associated G protein that prevented the release of NK cells. The mutation prevented the release of NK cells from the bone marrow, so it was involved in their maturation in some way. Also, a mutation called warm flash was picked up, and this was in the FLT3 receptor. This axis is required for maturation of conventional dendritic cells, and without it, the mouse will not survive. And a mutation called Gemini prevented CD8-positive dendritic cells from forming at all, and those mice too didn't fare well when they were infected. Finally, a mutation called Electra was tracked to Schlafen-2, and it implicated an entirely new class of cells, the inflammatory monocytes, in protection against infection. So this is the general high-altitude picture of how a mouse lives when it's infected. Now, could there be more proteins involved with non-redundant function? Very definitely, yes. We did cover a large fraction of the genome, but we weren't totally sensitive in our screening, I'm sure, and that's another factor that has to be taken into account. The last simple question we asked had to do with how the adaptive immune response occurs. And we started here with a lot of background. I think most people would agree that, first of all, you have to have T cells, and they come from the thymus. You have to have B cells of bone marrow origin, mainly, also from the fetal liver. And these the T cells interact with dendritic cells that send antigen. They proliferate, and then at the TB border, there's collaboration with follicular B cells to drive a T-dependent B cell response. And from there, you can have an extrafollicular plasma cell response, or B cells can alternatively go to the germinal center, undergo affinity maturation, and make high-affinity plasma cells or memory B cells. And our screen would test any part of this sequence of events. We also looked for T-independent B cell responses. The screen was carried out by Carrie Arnold, and it was quite simple. We took a Semliki forest virus vector that encoded a model antigen, beta-galactosidase. We injected mice with that on day 0, and this is a single-cycle infection vector that will infect cells, but it won't proliferate in them. It will infect only 2 million cells at max if we give 2 million PFU, and they make the antigen and they then die, and that's the source of the immunogen. On day 10, we would give NP-Ficol as a T-independent challenge, and on day 14, we'd bleed the mice and do a serum ELISA to look for beta-gal-specific IgG or NP-specific IgM. And we would also do flow cytometry on the blood to look for developmental problems that could easily be picked up. In all, 7,343 mice were checked for the T-dependent response, and of those, 6,198 were also checked for the T-independent response, and of those, 3,815 were checked by flow cytometry, since these were implemented at different stages in the screen. We had 31 hits, and of those, 26 were confirmed as transmissible, and 19 mutations were actually found, and they fell into 17 genes. I'll show you those genes in just a moment, but let me just tell you one more thing about the Semliki forest virus challenge. First of all, you see it definitely is T-dependent. If you have these are reversed. If you have no CD3 epsilon, you get no response. On the other hand, there's no requirement for either type 1 or type 2 interferon signaling, which should be here, and there's no requirement at all for MID88 or TRIF, which means it's TLR-independent challenge. Furthermore, one does get a response in splenectomized mice, and this is important because there are some mutations that you'll see that prevent marginal zone B cells from forming. They are not critical for the response to Semliki forest virus vector. So here were the genes that we found. You see that these are all the mutations in the left-hand column, and we were able to track down the gene in many cases. In those shown in yellow, we're still mapping at this time. One of the mutations was actually lost, but most of them were found by this point. And 5 of them were quite interesting, and those 5 fell into 4 genes. I won't talk today about empty hive and spelling, except to mention that these are required for follicular B-cell formation, but only in a context-specific way. The mutations prevented the formation of follicular B-cells in the bone marrow, and the mutation was tracked to ATP11C, which is an aminophospholipid flippase. Chris Goodnow and his colleagues found 2 other mutations, and it still is quite a mystery just why this flippase is needed for B-cell ontogeny. Another mutation called worker I will talk about. I'll speak about bumble and cellophane as well, and I'll tell you also about SCAN-T, which wasn't found in this screen, but it would have been had that formerly been part of the screen. It was just found rather casually by Owen Siggs, another member of the lab, who did FACS on a series of mice of his own. I'll talk about that one first, because this is a new protein that no one knew from human diseases that's essential for lymphoid and especially T-cell development. The mutation was picked up because in the mutants there was a pronounced deficiency of T-cells in the blood, also in the thymus, where, as you can see, there are only CD4 negative, CD8 negative cells. And if one looks at that population, overall the appearance of these mice is rather similar to a conditional knockout of Notch in its appearance. So it's something that affects T-cell ontogeny very early on. Looking again at the blood of these mice, one can see that there's a selective problem with T-cell development, but little, if any, problem with B-cell development, NK cells, or all of the other formed elements in the blood. On the other hand, if lymphopoiesis is examined under competitive conditions, one finds that B cells and NK cells are also dramatically affected. So it just depends how the phenotypic assay is done. This mutation does affect all lymphoid classes, but it doesn't affect myeloid development. Looking at the Hardy fraction of B-cell development in the bone marrow, we see that the defect becomes apparent about at Hardy fraction C, and thereafter it's maintained. And in the thymus, it's early after the double negative 1 stage that there's a clear defect, and that persists out farther. This mutation was tracked down in the usual way by bulk segregation analysis, and it was found to be a point mutation in a gene called ZBTB1. The BTB motif comes from the fly world, and it stands for bric-a-brac tram-track broad complex, and this is one member of a rather large family of proteins, all of which are transcription factors. We rescued the mutation by transgenesis, but didn't really need to do so, because on rare occasions we would find that the mice showed reversion of the phenotype. Here's the typical phenotype. As you can see, there are no T cells, and in a mutant here we find no CD3 positive cells, but in a very few mice we did see CD3 positive cells, and when we looked at tail DNA, they were indeed mutants. You can see here a C instead of a T at the critical residue. If we looked at B cells, we also found that the mutation was present. But if we looked at the new B cells, we see that there had been reversion from a C to an A. And at the protein level, wild type at that position is a cysteine residue. In the mutant, we have an arginine, and in the revertant, always we would see a serine rather than the wild type cysteine, but evidently that was close enough in structure that the protein would be rescued, and the mice would develop T cells that would expand given their proliferative capacities. The ontogeny of these proteins in the immediate close group of the family is shown here, and these are all proteins that have multiple zinc finger motifs as well as the N-terminal BTB motif. And if we look in the tree of ontogeny in the thymus and periphery, we know already of some proteins like LRF and TH-POC, which is its close relative, MAZR, BCL6. All of these proteins are involved in distal elements of the T cell tree. ZBTB1 is required very early on, either for T cell development near the point of the common lymphoid progenitor, or for B cell development and NK and PDC development too, if you define things based on a competitive assay. So here we have a new member of the family, and it affected ontogeny, but not all of our mutations had such gross effects. Bumble, by contrast, was a problem with distal B-cell ontogeny, and it was much more subtle. It was not picked up by FACS analysis, but simply by immunization. And it was picked up, as you see, both in T-independent assay and in a T-dependent assay. In both cases, there was a failure to respond to immunization. The mapping was performed using the T-independent challenge, simply because it was quicker. So here we had the affected population of F2 mice, and these were unaffected. And bulk segregation analysis revealed that the mutation was on chromosome 7, a 9.7 meg critical region was established, and when whole genome sequencing was performed, only one mutation was found in that interval. That was in a protein encoded by the gene NFKBID, and this protein is known as IKBNS. The protein had a splicing mutation, as shown here, and we found that we could detect the protein with an antibody after induction with anti-IgM antibody in the wild type, but there was no visible protein produced in the case of the Bumble mutant. Only anti-IgM and not other stimuli that would activate B cells, like anti-CD40 or LPS or CpG, was a strong driver of the induction of this gene. And induction of the gene also depended on the IKB kinase complex, because a NEMO mutant that we had in our collection, called PANR2, would prevent IgM-stimulated induction of the gene. Therefore, it seems that the BCR somehow signals to the IKK complex. Probably through NF-kappa-B, it induces NF-kBID gene. This, too, is a member of a family of proteins. Everyone is familiar with the conventional I-kappa-B proteins that limit translocation of NF-kappa-B to the nucleus. But the three novel or unconventional I-kappa-B-like proteins include BCL3, I-kappa-B zeta, and I-kappa-BNS. And these two had known immunologic connections at the time, where this one really did not. What we could say about the phenotype here was that, first of all, T-cell development was normal in Bumble mice. Furthermore, B-cell development was normal in the bone marrow of these mice. But marginal zone B-cell and B1 B-cell development were impaired in Bumble mice. And you can see there are practically no B1A cells and a reduced number of B1 B cells. This tail represents marginal zone B cells, and it too is reduced. So to summarize all of those data, these were the formed element problems in the B-cell animals, with B cells in these animals. Everything else seemed to develop fairly normally. We wondered if the lack of marginal zone B cells was the problem that caused the defective humoral response. But we thought later probably not, because splenectomized mice, as I showed you already, have a normal response to RSFV, and also the defect was intrinsic to bone marrow-derived stem cells. In fact, CD4 cells also are depleted, and B cells really quite markedly depleted if one establishes a competitive situation, transplanting B cells from bumble and wild type in a mixed form into a host. And marginal zone B cells are almost completely gone under those conditions. It was in such a mixed chimera that we did an immunization experiment with a T-dependent antigen, NP chicken gamma globulin, and we were able to look at haptenated B cells from the chimera and then ask what was the response of B cells of wild type versus bumble origin separately. When B cells are activated and when they begin to undergo affinity maturation, there's usually, or rather when they begin to undergo conversion to an extrafollicular plasma cell state, there's normally downregulation of B220, and that you see in the wild type population. About half the cells have undergone such downregulation. But you don't see it in the case of the bumble mutant. Here you find a paucity of B220 low cells, even with regard to the fact that there are fewer of these cells because of their disadvantage in the chimeric setting. On the other hand, we did see germinal center B cells of bumble origin on day 14 of the response, suggesting that they could go to the germinal center and undergo affinity maturation. Quantitating these data, we then see a disproportionate lack of antibody-secreting cells at day 7 and a fairly normal number of germinal center B cells by day 14. And going along with that, if we simply stain for IgM-producing plasma cells in the extrafollicular regions, we see in bumble that there just aren't any such cells. They're very abundant in the wild-type mouse, but not in the bumble homozygotes. We therefore place the lesion that bumble imparts somewhat here on the pathway. This protein seems to be required for the decision to make early extrafollicular antibody-secreting cells. Now, the last two mutants I'll tell you about very quickly because we know rather little about them. One of them was called cellophane, and it was picked up also in the immunization screen. It too had both dependent and T-independent problems in antibody production and a problem with IgD production as well. Just I'll summarize quickly. These mice had a small thymus with a high frequency of double negative and single positive T cells and with low double positive T cells. They had a low frequency of CD69 TCR beta positive cells, but nonetheless they had a normal T cell count in the spleen. They had almost no NK cells in the spleen. They had a deficiency of both marginal zone and B1B cells, rather like the case in bumble. But very much unlike bumble, they had fewer germinal center B cells than in normal mice, and they had defective affinity maturation when the mice were immunized with NP-CGG alum. This protein was known as ZEB1, and it's a member of a family that contains ZEB1 and ZEB2. It's known for its homeobox domain, also for a number of zinc fingers, and for both activating and repressing domains that are involved in the transcription of several targets. The knockout of this locus is universally perinatal lethal and has severe skeletal malformations. There's a milder allele known that has about 80% mortality within 2 days following birth, and probably for this reason no one suspected that the protein was involved in immunity at all. But the cellophane mutant has a normal lifespan. It's a premature stop codon mutation that cuts off this part of the molecule. Evidently, that's not required for life or for formation of the skeleton. From work done by developmental biologists, it's known that ZEB1 is involved in SMAD signaling, also involved in chromatin regulation, and we at present do not know how it causes the phenotype that we see. Finally, we had worker, which was a defect of T-cell production and T-dependent humoral responses, and it was picked up, as you see here, as a mutation that caused a low number of both CD4 and CD8 cells with a very high CD44 expression on CD8 cells, as though an attempt to compensate was being made. We found no problem with the NP-specific T-independent response, but there was a problem with the T-dependent response. Interestingly, these mice, first of all, if they showed the immune phenotype, all also showed a white belly spot. And here you see a row of these mice, all of them with rather large white belly spots. The mutation was dominant, and it was homozygous lethal. And so, too, here in this case, the knockout would not be expected to show a phenotype in a homozygous form. You'd simply have lethality. Only about half of the heterozygotes show an immune phenotype, but if you have an immune phenotype, there's 100% concordance with white belly spotting. Now, what does that mean? On the one hand, the spotting reflects a problem with melanocyte migration or melanoblast migration in the embryo. This is a cell type that's very different from any precursor of the hematopoietic system, or so we think. And furthermore, there's absolutely perfect concordance now in scores of mice between the phenotype and the white belly spot. All this suggests to us that there's an epigenetic effect of the mutation, and that's exercised probably at the level of the zygote. This mutant was established by the usual way of bulk segregation analysis, and we found it as a heterozygous defect of splicing in a protein called RUVBL2. And this too is a protein with two family members. There's RUVBL1, which has been crystallized and shown to have this beautiful hexameric structure. It's believed that RUVBL1 and RUVBL2 heterohexamers do form, and a lot of functions have been assigned to these two proteins. They're believed to be involved in chromatin remodeling and in the sliding of nucleosomes along chromatin. Also in the INO80 complex, they may be involved in homologous recombination or non-homologous end joining. In an assay that we did quite recently, we found that knockdown of RUVBL2 did affect homologous repair, but not non-homologous end joining, which is consistent with the specificity of the lesion for T cells, but not B cells. So where do we stand here now? What have we learned? Something very interesting, actually. I've told you about 5 genes now, actually. ATP11C, remember, is this gene I didn't talk about much. It's the aminophospholipid flippase needed for B-cell formation. Then we would put ZEB1 and RUVBL2 here. They're somehow needed for T-cell formation. NF-kappa BID is needed for extrafollicular plasma cells to form, and ZEB1 is definitely needed for affinity maturation. Those are the new genes that we found. The old genes could fill in the picture here. Also found by an unbiased approach, we know of DOC2, we have a mutation also in CD40 ligand, in MAP3K14, IRF4, Blink, and I won't go through the whole list, but all of these other hits have very well-established, well-studied functions. What we do not see is any mutation that has an effect on the dendritic cell compartment or on the innate immune phase of the adaptive immune response. In other words, we know of no mutation that will forbid the T-dependent immune response when some leaky forest virus vector is injected into the mouse. And we know now that we've covered a fairly substantial portion of the genome. So why is it that none of the mutations operate within the innate immune compartment? One possibility is that we're not at saturation yet, and such a mutation has yet to be found. Another might be that functional redundancy is an inherent property of the innate immune system. It's also possible that the adaptive immune system is just a lot more autonomous than we think, despite all of the nice drawings that we make, and we've exaggerated the importance of the innate component. It may be a combination of these reasons as well, or it could be another reason that I haven't thought of yet. And with that, I'm going to just close and thank the people who did this work. It was Owen Siggs who did some of the last mutations that I showed you. He was the one who found the ATP11C mutations and also SCAN-T, and Kerry Arnold carried out the formal screen that found most of the other mutations. That was my group at Scripps. There were several other people involved in finding MCMV mutations who had left the lab before that slide was taken, and they're all mentioned here. Karine Crozat began the MCMV screen and found about half of the mutations that I showed you. Our funding for this work was from NIAID and also the Bill and Melinda Gates Foundation for the MCMV screen and the immunization screen, respectively. And I've left Scripps now a little over a year ago, and this is my group as it's been reconstituted at UT Southwestern. And I think they're really the finest group I've ever had, and they're certainly more capable of finding mutations than the other group was, not only because of their own merits, but because the technology has improved a lot. And with that, I thank you very much.
We do have a microphone now, although we're running late. So if they're not burning questions, we'll release you, Bruce, and thank both speakers for a terrific session that closes Grand Challenges 2012 and opens the Keystone Symposium. So thank you.